Hi everybody, in this video we're going in eukaryotic cells. Now remember, this is cells with a nucleus, so that would include human cells. There's actually a processing that happens to the RNA after it's produced by that process of transcription. In our DNA, we actually have different regions. Some of our DNA is referred to as introns. This is non-coding sequences of DNA. We also have regions which are called exons. This is the exciting stuff that's expressed. So these are the coding regions that are eventually going to be used to produce a protein. So we can see an example of how RNA processing would work in eukaryotes. So here we have an example of a gene where there are exons, exon 1, exon 2, exon 3. There are also introns. Now, the intron sequences are going to be transcribed, but eventually those are going to be cut out, and the remaining exon pieces are going to get put together, and it's those pieces of RNA that are producing a finished messenger RNA that the ribosome is going to read in order to build a protein. Now, it's possible sometimes that the information from exon 1, exon 2, exon 3, it could get stitched together in different ways. So we might have all three together, and that messenger RNA goes to the ribosomes, and there's one specific protein that builds. Now, in a different situation, it could be that the information from exon 1 and exon 3 get put together. The information from exon 2 is not used. Now, because we've got a slightly different situation there, we can actually have the same gene here responsible for producing a different protein. And this is why, again, in the human organism that we have somewhere in the neighborhood of 30,000 different genes. This is why we can produce more than that number of different types of proteins. So for many of our genes, they actually produce not just one type of protein. One gene might produce 2 or 3 or 4 different kinds of proteins because of this different stitching together of those exon sequences. What's a mutation? This is when there's a change in the DNA sequence. It's a change in the ordering of those nucleotide base pairs. Remember the A's and T's and G's and C's. And it may result in a modified gene product, so a modified protein. It might actually result in a protein that should be built not being built by the cells that have that mutation. There are many different types of agents which can cause mutations. They would include things like X-rays or ultraviolet light, chemicals, and sometimes mutations can even be spontaneous. That there's just random times where the DNA is not copied correctly, and that causes there to be a change in the nucleotide base sequence. On this slide, we're seeing examples of different types of mutations. We're going to look first at examples of different types of point mutations, which involve just one or a few different nucleotides. We can have a base pair substitution. Maybe you were supposed to have an A and a T. After the mutation, you have a G and a C. A silent mutation, this is one which is not going to affect the protein product. So it may change the DNA sequence, but the end amino acid sequence in the protein is going to be the same as what was intended. A missense is going to completely change the amino acid sequence. Nonsense, we get the coding for an early stop codon, so we're not producing a protein that should be produced. We can have deletions and insertions. These could be 1 or 2 base pairs, which will lead to a frameshift mutation, which can be missense or nonsense. We can have a 3 base pair mutation, which is going to be a short missense, but it's not really going to be an extensive frameshift mutation in that case. Possible effects of mutation. It is possible that a mutation could alter the code so that no protein is going to be made. It might produce a different protein, which is non-functional. It doesn't do anything really for the cell. It might actually, mutation could lead to a protein whose function is altered and actually winds up doing something different than what the original version of the protein should have been doing. The Lac Operon is an example of gene regulation. Genes can actually sometimes be turned to the on position or to the off position. So here, the Lac Operon, this is present in many bacterial cells. Bacteria are capable of using different sugars as food sources. They can use glucose, they can use lactose. So in this first setting, what we're looking at is a situation where glucose is present, there is no lactose. This Lac Operon, there's a repressor, a protein molecule, which can hook onto the DNA, hook onto the gene, and it's going to block RNA polymerase. So then the gene is not going to be producing any protein product. Here's a different situation. We do not have glucose, but lactose is present. Now the lactose, when present, will attach onto that repressor. That means that the repressor is not going to bind onto the gene. So then the gene can actually be transcribed. This is actually then going to allow that cell to produce enzymes which allow it to utilize lactose as a food source. Now, if you think about it, for the cell, this makes a lot of sense. The cell is going to produce those enzymes that allow it to use lactose as a food when lactose is available. If lactose isn't available, then the cell's not going to waste its energy producing enzymes that it can't even use at that time anyway. In this slide, we're looking at some examples of ways that DNA can recombine naturally in cells. Plasmids are small loop-shaped pieces of DNA that are found in bacterial cells. So there's the normal genome of the bacterial cell. That's a much longer loop piece of DNA, winds up getting all bunched together. A plasmid is a small loop piece right here. Now it is possible that this DNA can transfer in between cells. In this case right here, we're seeing an example of conjugation, and this is where there's a structure that actually forms between this bacterial cell here and this one. It's called a pilus. It's basically a tube, and that tube, once formed, can allow this plasmid DNA, once replicated, to actually travel through the pilus and move from one cell to another cell. This can sometimes lead to the transmission of a new gene into a cell. So if this cell had a type of antibiotic resistance, if it's able to connect with another cell by a pilus and pass that plasmid DNA to this new cell, that cell would now gain a new trait. It would actually gain the ability to be resistant to the same antibiotic that this cell was resistant to. So this is an example of conjugation, which is allowing transfer of plasmid DNA from one bacterial cell to another bacterial cell. On this slide, we're looking at some additional ways in which DNA can be transferred from one bacterial cell to another bacterial cell. I don't have an image to go along with it, but transformation is a really significant way which DNA can move into bacterial cells. It's significant because this is what was going on in Griffith's experiment when the non-disease causing form of pneumococcus was transformed into the disease causing form. Bacteria can sometimes actually take up DNA from outside their cell, bring it inside, and if there are useful genes in there, they can actually put them to use. Now, Griffith's discovery of transformation, he didn't do a lot with it afterwards, but later Oswald Avery does additional experiments which actually help to show that DNA was the molecule responsible for heredity. Now, transduction is the third way in which genes can move from one type of bacterial cell to other bacterial cells. Bacteria can actually get infected by viruses just like our cells can. And in the process of transduction, it's actually possible for a phage, a bacteriophage, this is a virus which infects bacteria, to inject DNA into a bacterial cell. So it's actually possible to move new genes into bacteria by this process of transduction. All of these factors, new ways of moving genes into bacterial cells, is contributing to the process of antibiotic resistance. It's a very big challenge because we need to find ways to try to fight against microbes which are resistant to many of the drugs that we use to try to treat diseases caused by these microbes.